Ladies and gentlemen, good afternoon, good evening, wherever you are. My name is Sir Lambert Mbom. Welcome to this edition of COVID-19 in Africa. I'm sorry that I'm late today. We had some technical difficulties that kept us trying to fix this on the back. I apologize for this, and we'll try to improve on this next time. Today, we are going to be talking about COVID-19, Africa, and the vaccine controversy. June 20th comes one day after Americans celebrate what is called Juneteenth, celebrating the anniversary of the Emancipation Proclamation, which was done by Abraham Lincoln, proclaiming the freedom of slaves. In fact, when this happened, it took another 2 years for this to be realized in a particular city in the United States. It also comes on the heels of Father's Day. Tomorrow, we shall be celebrating Father's Day. But we can look at all this within the context of the events that are happening, because we are still in the pandemic, the COVID-19 pandemic. And because of that, we want to focus today on vaccines. Many controversies have arisen since the advent of the coronavirus. One of such controversies has been that of vaccines. There are many questions around vaccines. When shall we have a vaccine? What is the timeline for the first vaccine? We have heard 18 months is the time it takes to get a vaccine. Then we heard 6 months. But what really goes into a vaccine? And what about the famous HIV vaccine that they're still looking for? Even when the vaccines become available, when shall they get to Africa? If there is a cure, what is the need for a vaccine? And Africa often seems to be playing second fiddle. Remember when the world was searching for a vaccine for HIV, there seems to have been a breakthrough with Cameroonian-born Professor Anomangu of blessed memory that did not get any international recognition. The same seems to have come to life with the treatments for COVID, such as the Madagascar reality and that of Archbishop Cleda in Douala, among others. And we can also ask, how safe are vaccines? And even within the context of these, we still have other controversies that are coming up. And one name that has been floated and heavily criticized with respect to vaccines is that of American billionaire Bill Gates. With me today to walk through these questions is Dr. Jacob Minang. Dr. Jacob Minang is a research scientist with a master's degree in biochemistry and a PhD in immunology. He is the Chief Executive Officer of Optima Healthcare, Inc. Prior to joining Optima Healthcare, Inc., Jacob worked as a postdoctoral fellow on HIV/AIDS vaccine development at the National Cancer Institute in Frederick, Maryland. He has also served as a research scientist investigating immune responses to anthrax and seasonal flu vaccine components. Welcome, Doc.
Good morning, Mr. Lambert. It's a pleasure to be here.
So, Doc, I mean, we all have been hearing about this. Let's get this started. Please, what are vaccines in simple terms?
Okay, so the whole concept of vaccines is using parts or wholes of an organism to get the human body to have experience of that organism. You can have an immune response for the organism without having come in contact with the actual organism. That way you have some kind of a memory. That's the idea. And the thinking being that when you eventually encounter this organism for the first time, because you already have memory of that organism, you're going to have a better and a sustained immune response that will be able to prevent you from getting very sick or even potentially dying from an infection.
So, in essence, the most important thing we need to take away is that you create a memory so that when you come in contact, then you can the disease or whatever, you'll be able to recreate that so that you prevent it from replicating itself. Do I get that correct?
Absolutely. So stepping back from diseases and think only about the concept of memory in general. When you are teaching your children, you tell them things repeatedly over and over because you're hoping that they can keep it in memory. when you try to get that information back from them, the better their memory, or the more often they have kept that information, the more quickly they can give you that information. And even when they give you the information, the accuracy of that information depends now on how good their memory of that information is. That's how you distinguish between children. So what the vaccine is trying to do is, because usually when you get an infection, the body has receptors for different organisms, but these receptors are not as good as they should be. We are born with them, so they're called innate. But when you encounter an organism, then you can end up with an adaptive response, which means there's a response that's adapting to the fact that you have actually encountered an organism that helps to actually clean up the response and eventually generates a memory that is stored in your body. If you encounter the same organism a couple of years down the line, it's possible for you to clear the infection with very mild or no symptoms because of that memory, because you're able to have a very rapid and sustained response to such an infection. So that's the whole idea about vaccines.
So I realize, I mean, we can take this progressively, but one thing that I have been fascinated by is the fact that when we're kids, we're vaccinated against a couple of things, isn't it? Measles, polio, and all that. But when we became adults, I never heard anything about vaccine again. And until I wanted to travel, I started hearing something about vaccination against yellow fever. Is it that the memory we had has been stored in, and in case we come back, we have a polio attack or something later, it is already in our system for the rest of our lives? So how does that work? Why did we have that and not have it now?
Okay, so to step back a little bit, there are a lot more microbes out there than there are human beings. Just the species or different strains of microbes, not even the number in each species, but there are a lot more microbes out there than there are human beings. And a lot of these microbes, we can handle them without having any memory response. Just like innate responses in the sense that just your saliva, a lot of things that when you encounter through your mouth, you can take care of them by just the saliva. The mechanical barrier of your skin can take care of a lot of things. But there are some organisms that successfully break through those barriers. They break through the saliva barrier, the mucosal barrier in your lungs or your skin, and get into your body. And even some of those, a majority of them, the human body can over time eventually clear it. But there are some that can lead to debilitating diseases, especially in the childhood. And why you're talking about childhood and vaccination is because at the level of as a child, you don't have memory of having encountered any organism when you're born. But as you go out there into the world, you are flowing through a stream of millions and millions that are shaping your immune response. So when you are born as a child, you depend on your mother to have what is called passive immunity, because your mother has experiences with organism, your mother has antibodies against those different organisms in their bloodstream, and they pass that to you. So your first few months of life, you're totally dependent on that passive immunity, as opposed to adaptive immunity that you yourself have created over time through your own personal experiences with organisms. Excuse me. And that's why they encourage breastfeeding, because even postpartum, you can continue to benefit from passive immunity from your mother through the breast milk. Okay, but during childhood, again, now most vaccinations should happen at that age for the obvious reason that I'm talking about adaptive immunity. You want children to be exposed to particles or whole molecules of organisms that are dangerous that we know about, and we have vaccinations from them. That way, you have that memory as a child. Then, as you grow up, as you obviously pointed, you're never going to hear about vaccines again because you don't have again to be vaccinated. You've already been vaccinated as a child, so you are protected against those infections. So as you encounter those infections in the course of your life, your body is taking care of them. If you are given a vaccine that works, and if you are not immunocompromised. So the only concern, therefore, is those other organisms out there that, unfortunately, as we speak, we don't yet have vaccines against them. So obviously, you were not vaccinated against them when you were a kid because it's not vaccine. If there was a vaccine against HIV, you'd be given that vaccine as a kid, just like you're given the measles vaccine.
So, I mean, let me get into that deep, because the other thing that I'm wondering is we often heard about malaria, we heard about fever. We don't have vaccines for those? Is it because they are not compromising the immune system? Why is it that we are looking for vaccines only for things like this and not for malaria and the rest?
Okay, so that's a very interesting question, but let me point out a little difference. There's fever and there's malaria. Malaria is a disease that has a specific pathogen that causes it. Fever is a symptom of so many diseases. It's a manifestation, and so you can treat fever, which means you're treating a symptom, but you still have to do a diagnosis as to why is this person having fever. The fever could be due to the seasonal flu. It could be due to typhoid. It could be due to malaria. It could even be the early signs of HIV or nowadays corona. So just to separate that out. So then getting back to your question, malaria. Malaria is is of huge interest to me and is very near and dear to me because my master's degree thesis was on malaria. Actually, I work on the most potent malaria parasite that affects people in subtropical Africa, and as you can imagine, is more deadly among people below age 5 and pregnant women because pregnancy leads to a reduction in suppression of immunity because the child that you have in your womb as a mother is like a foreign body. It has antigens that are not yours, that are from the father. And in a sense, women can treat children in their body almost as an infection and potentially get rid of their own children. And that's why nature has made the immunity of a pregnant woman to be a little bit suppressed. And because of that, they can tend to be more susceptible to some type of infections, in this case, malaria. So I studied malaria in pregnant women and their newborns and the type of protection. So to answer your question, it's not true that there's no work on malaria vaccines. Just like with HIV, there's been a lot of work the last 100 years to get a vaccine for malaria. But the parasite that causes malaria, just like the virus that causes HIV, those are two very complicated organisms. They tend to change their nature. So we're going after an organism that changes so much that even whatever molecule from that organism uses as a vaccine, the next infection, that same molecule presents in a different form. The vaccine ends up not being as effective. So that has been a struggle with malaria and HIV. Unlike the other organisms will have vaccines for one thing, they might be that they don't change their form that more. For another thing, it might be that it was easy for us to use the whole organism for a vaccine. Like for smallpox, where they found out that milkmaids, people that milk cattle, that have cowpox, that's a different kind of virus, but it's pretty similar to the human one. When they get infected with cowpox, that causes less of a disease to those people. They end up being protected against the human smallpox. So the difficulty in getting it with the complexity of the organism and its ability to change its form.
That is interesting. You mentioned a couple of things there. What is the difference between an organism, a parasite, and because again, are they all a virus? What is it? Are they all the same?
Organism is a generic word to refer to anything that's living. So we humans are organisms. So after we've talked about parasites, we've talked about parasites are organisms that are living, that are dependent on other organisms for their own survival. So then they are parasitic, as opposed to where you can have symbiotic relationship where that organism is dependent on another for their living, but they're also contributing. Like our gut, our stomach are full of gazillions of microbes. A lot of those microbes we will not survive without them, but they too will not survive without us. Then we don't call them parasites. We're having a symbiotic relationship with them. Almost every organism has some organism that is dependent on it. Even viruses that we think that are very deadly, they have bacteria that are dependent on them, and they have a relationship that is called bacteriophage relationship. So, but when we have defined what an organism, be it a virus, a bacteria, or larger mammal, then we can continue the conversation by just referring to them as organism, because the generic word that just refers to something that's living.
So to backtrack to where we started off, vaccine is meant to create the memory, okay, so that you can, once it comes in contact, that memory can continue to play on and ward off whatever, isn't it? Now with the virus, the particular virus in case the coronavirus, what is this debate about the virus and where are we with that development?
Okay, so first off, a basic, very little is known about this virus so far, and anybody that's claiming to know a lot about the virus, it's somebody that is trying to sell you what do they call it, fish oil. Okay, so we are all learning about this virus. Where we are with this virus is where we were 40 years ago on HIV. Very little was known about HIV, so everything was speculation. The difference between this virus and HIV in terms of knowledge is not unique to this virus. It's about the body of knowledge about viruses in general so far, and also the body of knowledge about vaccines so far. And so when people are doing projections about how long they think is going to be, we're going to be able to get a vaccine, it's not so much about coronavirus, but about what developments and infrastructure have been built around developing vaccines in general for the last 40 to 50 years when vaccine research started. So we think we have enough technology so far that once we can be able to pinpoint a molecule in the virus that we think could be a good target for a vaccine, then we can very quickly scale up and produce the vaccine in large quantities. That's why people can be putting a timeline. Unlike with HIV 40 years ago, you couldn't put a timeline because even the vaccine research itself was at its infancy. It was very little about biotechnology. The primary reaction was just 10 or a couple or 2 decades old. The idea of being able to take a molecule of DNA and increase it in large enough quantities that you can be able to go back and sequence and tell what organism it comes from and what protein it can produce. All those were new technologies. So we have technologies now that are not organism specific. So that with new and emerging infections, there's a lot more we can do. But you still have to do that legwork of studying and understanding that organism itself and how quickly it changes its form. And so the struggle now among the 200 candidates of vaccines is being able to pinpoint the organism that will be the best target. Because as far as the technology and the platforms to rapidly manufacture a vaccine is concerned, we don't have a problem with that. Those technologies are already out there.
So just walk me through. I mean, as a scientist, the virus has been discovered. Now what is that process of looking for a vaccine? What does that look like concretely?
Okay, so a quick reminder. First of all, a virus in the true sense of the word is not a living organism. Unlike a bacteria, a protozoa, and most other organisms, because when you have a bacteria in a droplet lying on the surface like a doorknob, in there it can't do anything. If there are no other living cells like mammalian cells, it's just in it. It's like a piece of metal, basically. The only difference is that it has message, it has information in it that it can use to replicate if it ends up in a living cell. And now, if in a living cell for an organism in which it can survive, and that message is that genetic material is called RNA. Okay, so the RNA of itself is essentially like any other innate object, but for the fact that it has that message, so that's the first basis. So what happens then is when you touch the dog that has that or any surface, or you shake somebody's hand, or you're passing through a space where somebody has been coughing and you have the droplets there and you inhale it, depending on how much of that you inhale and it gets either through your nose or your mouth and ends up in your lungs, the virus can eventually find itself by binding to a receptor on a cell into a cell. And it needs to get that message all the way to the nucleus of that cell. And that message now we're able to integrate into the genome of that cell. It's only at that point that you can describe the virus as being living, because at that point, it's gonna overtake the host genetic material, the host genome, and take advantage of it to use to be able to produce its own progeny, its young ones. I think Prof just joined us.
Yes,
and then you have particles being produced, tons and tons of them. That can lead to now more cells in the body of that individual being infected, and that individual also is spitting out large quantities of the virus into their mucus that they can now be able to go around and be transmitting to from one person to the other. So that's basically the background. So when you have a new virus then and you're having infection, what happens is you start from the fact that a lot of people are being sick in the hospital. Then you walk back to the cause of that infection. And the first thing they notice is that when you're doing diagnosis using the common organisms that are known to cause those type of symptoms. And we have gone through those array of organisms, and none of them seem to be turning out positive. That's when you then try to get that message from the blood sample or mucus sample, and then increase its quantity using the polymerase chain reaction that I just mentioned to you. That was only a new technology back when HIV was discovered. But now, within a few short days, they can quickly sequence it to a large quantity and be able to now link it to an organism that we did not know about. And most of the time, it could be a new organism, but we're relative of other organisms that already exist. Like in this case, we're calling it corona, but corona is a family of viruses. So we already know about coronaviruses, but we don't know about this specific one. Just like we know about human beings, but we might not know about Eskimos, or we don't know about black people. You get what I'm saying?
Right.
Yeah. So we're able to, we know that it belongs to a certain family, and we know that the viruses of that family tend to be respiratory viruses.
So, Doc, again, you have said quite a lot. We want to welcome Professor Kenneth and Chang all the way from Nigeria. Prof, can you hear us?
Yes, I can hear you. Can you see me? No, not quite, I'm sure.
Yes, I think you need to turn on.
But you don't have your video on.
Yeah, you need to start it. Let me put your name, you can come on again. So, Doc, I wanted you to walk me through again, because you talked about the RNA, the receptacle, and then getting into the nucleus of the cell. When it gets to that nucleus of the cell, then it spreads all across, isn't it? So when you talk about the vaccine, what particularly are you targeting?
Exactly. So you have vaccines and you have drugs. Okay. The Holy Grail is a vaccine, because a vaccine will be able to stop an infection in its tracks. But in the absence of a vaccine, then you have a drug then that will tend to target biological functions in the virus, like maybe an enzyme in the virus that helps the virus to replicate. And if you can inhibit that enzyme with a drug, then you're controlling the spread of the virus, the multiplication of the virus in the person, which is what we do now with HIV. We have drugs, but we don't have the Holy Grail that can even prevent infection, which is the vaccine. So a vaccine is looking at targets on that virus when it's just particles and it's lying there. If you pick it up, you want to have a response that blocks the entry of the virus into the cell. That's the ideal vaccine. A vaccine that either will generate antibodies that can target the virus and kill it when it's still free floating in the bloodstream or in the mucus, or prevent it from binding to a cell, or will kill infected cells, because those infected cells are expressing parts of the virus on their surface, and a T cell can see those infected cells and attack them and kills them.
So too much information there for us, Doc. So again, you just made a distinction between a drug and a vaccine. Your best bet is always to get the vaccine, because the vaccine it prevents it from you prevent the infection before you start looking for the drug and all that. Am I right?
Right.
So what is it the vaccine is targeting precisely? You already explained to us here that when the virus gets into the system through whatever means, through the droplets and all that, it is targeting a particular cell.
Right.
So why is it so difficult? You have already identified the cell. That is, why is it so difficult then to get some?
Okay, so thank you. That is a very brilliant question. And that's why I told you from the very beginning that anybody that is claiming to know a lot about this virus at this point is trying to sell you snake oil. Okay? Because you said you know a particular cell. Nobody knows a particular cell. People are guessing different cells. They're guessing that since it's a respiratory virus, it's respiratory infection, then you might be infected. The infection might be going through epithelial cells. But even if we know what cell it is, you have gazillion receptors on cells that virus could potentially be binding on. So if you confirm what cell it is, some virus can multiple cells. Like the HIV virus, everybody used to think that they infect only CD4 cells, T cells, but they infect more than those. Everybody used to think early on they thought it uses only the CD4 receptor entry. But there are so many receptors that use it. And so it tends to infect a lot more cells, even though some cells are their major targets. So you might start by knowing the major target of a virus, and then you tend to zoom in on that. You need to realize that despite the fact that you have brought in all the things that you need to prevent it from infecting that particular type of cell, people are still getting infected. That's how you realize that there are other minor targets that you might have. There might be a major target. The major targets usually are easy to define. But eventually, over time, you realize that either there were already some minor targets at the beginning, or because the virus has the ability to change its form very frequently, it's been able to adapt to infecting some other minor targets. So first, there's the target. Knowing what the target cell is. The next thing is, what are the molecules on the target cell that the virus is using to enter? And then on the virus side, what is the virus using to be able to attach itself to the target cell?
So, Prof, can you hear us?
Yes, I can hear you very well. I can hear both of you very well. Yeah.
I don't know what is wrong with your video. We can't see you. We're only seeing your UCAM.
Yeah, that's right. I can't. I'm also trying to figure out what's going on here. Let me see. Just a minute. Okay, the camera. Okay, sorry, but you can write on. It's okay. I think while you are on, I would figure out what I need to do. It shows here that the camera is on, but I don't know why I'm not. Let me see.
It says,
okay, but you can write on.
So maybe you have to click a start button on your cam. It says,
maybe
please
try.
Okay, now, yeah,
okay.
let's see.
Okay, Doc, while you're Prof, while you're trying to figure that out, there are some questions for you, Dr. Minang. Dr. Nicholas Starr is asking, what is the difference between a virus and a bacteria? What's the difference between a vaccine and an antibiotic? And can an antibiotic be used to cure a virus? And can a vaccine be used to cure a bacteria? Very interesting questions. Thanks, Dr. Nicholas Starr.
Okay, so those are very good questions. So I'll take them one by one. That way I don't forget anyone. The first one is the difference between a virus and a bacteria. Remember, at some point I told you that a virus literally is innate. It's not alive. It's not a living organism unless it's in another cell. Okay, so the difference between a virus and a bacteria basically boils down in that what I've just pointed out. Bacteria are living, even though some of them need to be inside a cell, and some of them can live extracellular, but you have a lot more bacteria that are extracellular. They float around in your bloodstream, and they cause septicemia. You know, they're devastating in and of themselves, the way they are floating in the bloodstream, and then you talk about septicemia, which means an infection of the bloodstream, and you need to take antibiotics to clear them. An antibiotic is called as such because antibiotics specifically target bacteria. Okay, but what targets a virus, a drug has to be an antiviral agent. So that's the difference right there. And then the next question was about
can a vaccine be used to cure a bacteria?
Okay, so when you use the word cure, think drugs. When you use the word vaccine, think stimulating immune responses. So a vaccine can be used. You can have vaccines against bacteria, but the intention of that vaccine then is not to cure, because curing, you mean that you already have an infection and you're trying to control it. The intention of all vaccines is to preempt an infection in the first place. So vaccines are usually the only there because they prevent infections. Drugs are your last resort because you control an infection. And sometimes a drug might not be, and the beauty about drugs is that they don't necessarily have to be targeting the organism of interest. They don't have to be, for example, hydroxychloroquine is talked a lot about. It's not an antiviral agent. But it could have an effect against corona because it might control the symptoms. They might an environment that virus it thrives in. And if you use an anti-inflammatory agent, then it takes away that environment and the virus cannot thrive. So you're not targeting the virus directly, but you're creating an environment that is not conducive for the virus. So that's the beauty about drugs, because with drugs, you can either target the organism directly, or you can use a drug that creates an environment that is uncomfortable for the organism.
Yeah, Prof. I mean, I think we can do without the video. Can you hear us?
Very well. I can hear you very well.
Okay, I want to thank you very much, Prof. You have been patiently waiting for 2 hours to come on. Professor Kenneth and Chang Yongabi is in Nigeria and is currently the African, let me get that correctly, African Scientific Research and Innovation Council. This is coordinator of that, coordinating the Afrocentric response strategy to COVID-19 in Africa. Did I get that correct?
Yes, please. It's okay. Under the African Union aegis. Yeah,
Good.
Aegis, yeah.
So we are dealing with somebody who's working within the African Union and trying to coordinate an Afrocentric response to COVID-19. So, Prof, tell us how far are we with that?
Okay. Before I go on, first I'd like to thank you for bringing me once again on this your program, and then I also like to thank Dr. Jacob Minang for at least I've missed most of your talk, but I'm able to capture the last part of it, which is really, really good because you've been dealing with immunology and immunochemistry, and of course immunopathology, so which is really great. So I'm really happy about that. But before I go on to talk about myself, I just like to, if you would permit me, Dr. Jacob, to throw a few words on the question, on the answers that you've provided. I just want to take on, what is an antibiotic? I just want to take that before I come in to say more about what I'm doing, because I think that's very fundamental. What is an antibiotic?
Yeah,
can an antibiotic? No, sorry. I just want to be. I'm just following up. You've actually answered that. I just want to give some, throw some more light on that.
Yes, please, go ahead.
Now, for the audience, and to follow up what Dr. Jacob has said, if you break the word antibiotic into two, it brings the word anti and then biotic, something against life. All right, that's it. That's the very basic way of looking at it. So yes, antiviral agents are antibiotics. That's in that sense. Antiviral agents are antibiotics in that literary sense of the word. But then most of the time when they mention antibiotics, they are referring to mostly bacteria. But then, of course, you have antifungal antibiotics. You have antiviral antibiotics. So that's the meaning of the word antibiotic, the broad sense of it. But then, of course, you have the contextual definition. Then you have the conceptual definition. All right. So, and then you also have the experimental definition of it. So I just wanted my audience to take care of that. That when we say antibiotics, generally the word antibiotics today came from the what? Even came from bacteria themselves. Filtrates of bacteria themselves against other bacteria that causes diseases. That's the genesis of the word antibiotics. All right. I'm sure Dr. Minang, I'm just trying to get our audience to really get that very framework of it. So you have antiviral antibiotics as well. Then you have words like antibiosis, against life, So then you have antimicrobial antibiotics. That means, they are not from filtrates of bacteria, but they are coming from plant origin. So we now call them antimicrobials. I hope probably, Doctor, you agree with me on that. Again, so I just wanted to be able to throw more light on that. But Doctor has actually explained that. I want our audience to be able to, get that details. And then he says, vaccines, are they used for treatment? He has answered that question very exceptionally well. But I like to say that we would say vaccines prevent. I mean, I want our audience to get these words, prevent. He has explained it using the, I would call it the immunological jargons of it. But vaccines prevent. But in some rare occasions, you have what we call therapeutic vaccines. If you remember what Professor Victor Anomangu was doing, we would have classified that as therapeutic vaccines because it goes a little bit against the conceptual framework of what vaccines are supposed to be in their very entity in time and space. So I'm just giving that again for a conceptual background for us to be able to get it straight. So thank you, Dr. Jacob. That was just a follow up on that.
Thanks. That was very nice and very supplemental. And I like the last mention that you made of the therapeutic vaccines because at the introduction, our host Lambert was talking about the controversy with Dr. Anomangu, and that goes to the heart of that controversy because he was getting into a realm that was pretty new back then, the concept of therapeutic vaccines, which goes against the whole idea of vaccines that are supposed to be preventive. But yes, you can use a vaccine as a therapeutic. It's a little bit confusing where you're trying to get an organism from the same person that's already infected and is suffering from the infection, and then mess up with it a little bit in vitro, and then give it back to that person and hope to boost their immune response to the organism, which was the concept of his own vaccine.
Thank you very much. Okay, so back to what the host has asked me, where we are with this. It may be very interesting for our audience and our experts across the continent to know that the African Union Scientific Research and Innovation Council is made up of more than 200 scientists with their different expertise in their own rights. All right, so I was admitted into this council in 2018, and just 2 months ago, I was, of course, appointed to chair an inter-regional committee on the African Union Scientific Research and Innovation Council to coordinate an African response strategy for fighting COVID-19. That's the mandate. And this brings on board members representing scientists representing East Africa, scientists representing North Africa, scientists representing West Africa, scientists representing Central Africa, and Southern Africa as well. So it's really interregional. So I'm privileged to be coordinating this under the aegis of the African Union Scientific Research and Innovation Council. Now, what are the mandates? The mandates, what are the terms of reference, is to review all the strategies that have been carried out on the continent to fight COVID, with the understanding that some of these strategies, or most if not all, cannot be copied from Europe, America, and other Western nations because of the unique ethnographic, cultural, and strategic issues that Africa actually faces. So we can't be copy and paste. So what they are using as prototypes for controlling coronavirus or COVID-19 across the other continents can seriously not agree in time and space with what is obtainable on ground in Africa. So that is one of the issues that this working group is going to be looking at. Of course, we've actually begun our work. Now, in all the areas, in terms of drug discovery, in terms of vaccines, in terms of using non-pharmaceutical intervention strategies, of course, like you talk about physical distancing, can physical distancing in the US, can that work in Africa? Can water and sanitation issues, the way they have said it, the way WHO has put it, does it work? If it's working in Brussels, in Belgium, for instance, can it work in Nigeria? Can that work the same way in Cameroon? So this, consortium is to be able to look at all these intrinsic differences and see a strategic framework that things can work within the Afrocentric reasoning and the challenges that we have. That's that. Now, the committee and the working group also recognizes the fact that it is not easy to quickly develop vaccines across Africa. And let me ask this, is there any vaccine over time that African scientists have discovered on the continent that is being used globally? Ask that question right now. Let's think about that. Is there any vaccine? Dr. Jacob, you are there. Is there any vaccine that has been developed by Africans on the African continent that is being used globally, starting from polio, poliomyelitis vaccine? Is it developed from Africa? Smallpox, was that developed from Africa? Even you talk about typhoid vaccine, BCG vaccine for TB, was that developed in Africa? So we have been receiving. Right. We have been receiving. So these are the considerations that we want to be able to talk about. It's not like African scientists don't have the leverage to be able to develop this. But Dr. Jacob, you are in the US, probably you are abroad, for instance. If you develop a vaccine there, yeah, you're an African, but you develop it in the Eurocentric or the Western-centric atmosphere, the Western-centric thinking. It's not within the Afrocentric thinking. So these are the issues. These are the issues that we want to be able to look at. The immunology remains the same. The pathology remains the same. The science remains the same. But have there been, when we talk about vaccines in Africa, have there been vaccines? I know Professor Anomangu developed a vaccine, but which predominantly he did that in Europe, but was not even acceptable on the African continent. I just want to throw a little bit light on that. So I'll come in again to be able to go into some intrinsics. Thank you.
Yeah, but Prof, before I let you go off on that, I mean, can there be some African science that is opposed to Western science? Science is supposed to be universal, and wherever it operates, isn't it? Are you trying to?
Let me comment on that. So, Prof's questions are on point, but I think the frustration of the African scientists it's not about science per se, because as Prof told you, the immunology is the same, the pathology is the same, and the African scientists is equally capable. I think the problem is the politics, marrying science and politics in Africa, and the leadership that we have in Africa, because there's no reason why we shouldn't have the infrastructure to do our vaccines in a couple of major sites in Africa. It does not have to be every African country doing it. We can have a vaccine site in South Africa, for example, in Nigeria, in Ghana, in Egypt, and then they work as a consortium. The problem is the leadership that we have in Africa, and this their dependence on the West. Because when you even come to testing the vaccines, sometimes you don't understand the politics of how they decide to come and test a particular vaccine in a particular African country. It's more about money changing hands than them focusing on the rigors of the science, the regulations, and the consenting that's required to be able to do a vaccine study. In particular, remember the hoopla that we had about the French scientists talking offhandedly how we were going to come and test a coronavirus vaccine in Africa, even when we did not even have any infections in Africa at that time.
Yeah, but Doc, I mean,
okay, let me. Can I take a follow up?
Go ahead, Prof.
Okay.
Following from the discussion, your points that you've raised, Dr. Jacob, which are very great. One of the issues, I picked the last part of your discussion when I just signed on, where you were explaining about looking at the strains. We look at genomic sequences of COVID or viruses, particularly talking about COVID. Now, when you look at the COVID-19 across Africa, there are a couple of strains. It's not just one strain. You find strains that are phylogenetically related to the strains in Wuhan. You find strains that are genetically related to the strains in Italy. You find strains that are genetically related to the strains in America, in New York, for instance. Now, what kind of vaccines are you going to produce right now to be able to address that? That would bring about the genome, one peptide that has the signals or the genetic information for all the strains. That's the kind of vaccine we'll be looking at, especially when you're now having to give it to the whole population. Okay, because you're not going to now start testing different populations and say, okay, we have vaccine A, which is because you have Wuhan strain, we're going to give you vaccine A or vaccine B. So we look at the genomic sequences, you realize that, like you said, the COVID-19 is evolving a lot, and we're learning on the job. Scientists are learning on the job to be able to understand a lot about the genetics, the makeup, and things like that. So it also tells you that vaccine, it's still in the tunnel. It's still far away off to have an effective vaccine on the continent that would really respond and meet up all what it takes to be able to prevent COVID-19. It's still a journey to be able to make that comment. So vaccine, yes, the science is the same. Pathology, immunology is the same, but strains from Africa would differ. The genetic makeup of Africans in terms of what would differ. The health, situation of Africans, of course, you will look at the clinical situation of Africans. The comorbidities, the underlying factors will also differ. Socioeconomic determinants will also differ. So when you need to put about that's the Afrocentrism, and then the cultural issues would differ. Okay, because you remember when they are doing this mass vaccination in the northern Nigeria, the Emirate of Kano rejected vaccination of polio in the Emirate of Kano in Nigeria. Why? For cultural reason. They said that they are trying to sterilize their population from giving birth. So there are so many. So that's what I mean by the Afrocentrism. We need to be able to key in all these perceptions in order for us to move forward. Thank you.
So, Dr. Minang, Thank you very much, Prof. Let's get back to, where are we with the vaccine? Which are the most prominent ones that have come in, at least here in the US or across? Where are we with that?
Okay. Yeah. So before I answer that, I just want to add a little thing to what Prof said, which it was very brilliant. The Prof talking about the different strains and the genomic differences, and to kind of agree with Prof that that's exactly why we are where we are with HIV. Only in Cameroon, there are so many significant strains. There's a lot more diversity in HIV in Cameroon alone than in the rest of the world. So it means you actually have to get a vaccine that's specific or unique to just Cameroon before you look at other African countries. But another thing that scientists have been trying, which probably agree with me, is that there's something that is called conserved sequences. When you which means that there are some sequences that are the same across the board, no matter strains of the virus you're looking at. The question now is, are these conserved sequences the best targets that can give you the best immune response? How important are these conserved sequences to the virus? Sometimes it looks like those conserved sequences are because they are so important to the virus. Whenever the virus tries to make a change in those sequences, the strains that have those changes tend not to survive. And so the only strains that have those conserved sequences stay. But the problem is if those conserved sequences can be the best target, then you can be able to get a universal vaccine. So the holy grail for viruses that tend to change their nature a lot is to use the conserved sequences as your target for a vaccine. But again, the location of those conserved sequences might be in areas where the immune system cannot access them. Okay, so then let's get back now to the state of the art in vaccine research on corona. As I said, most of the hype has to do with the technologies around being able to scale up a vaccine once we nail down a candidate that is very good. But as far as understanding the virus and having a target that we show that is the best target, and before you start talking about universal targets, as I just mentioned about conserved ones, we're very far from that. So the talk about 8 months, 6 months, 18 months, and all of that is hoping that we can learn very quickly about the virus and come up with targets that we can use, then we can quickly scale. For now, there are about 200 candidates in the pipeline. But those are just targets that they're thinking that we can get at. And those are targets on the virus. But there are so many other steps above a target on the virus. You have to determine in what form are you going to deliver that targets to humans vaccination. Are you going to deliver that target in the protein form, in the RNA form, or the RNA form? And the next thing, what components are you going to, what the structure, what are you going to build that target around? And in immunology, we call this the immunologist's dirty secret, which is it's the word they call adjuvant. We know that word, but the whole idea of that platform in which you're bringing the target is that it can help to both boost the response beyond what you get if you are just giving only that target, but can also extend the response by creating more of a depot effect at the site where you have injected the vaccine, so that the actual molecule that is of interest is being released gradually over time to continue to boost the response absent the infection, so that by the time the infection comes, you have a stronger memory. So first, you have a target. Second, you have to have a platform, a delivery mechanism, which is what distinguishes the different companies that are in competition with the 1,200 candidates. An example is Moderna. For Moderna, their own platform is a messenger RNA platform. There are some other people whose platforms is a DNA platform. And then some are using the whole organism platform, which means that they are using attenuated organisms like adenovirus, and then which human beings have long experience with, and we control it, and they're attaching a sequence of the virus to it. And then you have to deal now with the third level. What is the best mode of delivery of a vaccine when we eventually get it to get the best response and the durable response? Are you going to give it IV directly through the bloodstream? Are you going to give it intramuscularly? Or are you going to give it as a droplet through the nose or the mouth? Or those different routes will end up with different outcomes in terms of either even getting a response or getting a durable response. As of now, what we hear is the few people that have been infected that cleared the virus, that in this 5 months of human experience with the virus, who have gone back and tested their immune response, they are realizing that the antibody levels wane within 2 to 3 months. They're no longer there. But that's looking all at the antibody levels. We have to look at the T cell memory, B cell memory, and they're still far from going. There are two, I think, they're promising because at least the immunology studies in monkeys and lower mammals and seen very strong response. They have done toxicity studies in humans.
Your line is
sorry
breaking for me now.
Okay, can you hear me now?
Good, yes.
Yes. So there are 12 candidates instead of the about 12. I start promising I've done at least 2 things with those 12. I found that they can elicit immune responses in lower mammals like monkeys, and others. They are very confident that they are not toxic in one of.
Okay, no, let me. I think there's a problem with your network there. Let me get to Prof, then come back to you. Okay. Just check your network because it keeps going in and out. But Prof, I'm sure you followed up the discussions with Dr. Minang, and he gave us 3 levels, and he talked about the RNA, the DNA, then you get over to best mode of delivery and all that. But I know that, and was something that he mentioned, which I know you have gone through, is he talked about boosting the system. Are you producing something called STK boosters and STK phytoimmune for COVID-19? How are these related to the search for vaccine, and what are these in precisely, Prof?
Well, yeah, thanks for that. That's very important. And the STK boosters and STK phytoimmune, what they essentially do is to stimulate the immune cells. I'm sure Dr. Jacob would begin to understand what stimulation of immune cells are. If you stimulate the immune cells and at the same time you boost the immune system, because it's immunostimulatory, you boost the immune system. How do you boost the immune system? And what are the immune cells? You have T cells, like he has actually mentioned. All right, there's T cell lymphocytes and all that. You have the macrophages. All right. So when you are able to boost these immune cells, the immune cells, what they do is that when they fight the infection, they are able to capture the infection, they neutralize the infection. There are very specific mechanisms in which this happens in the system. Now, in a nutshell, now it's not like we have taken the entire STK, formula and tested to what it does. We have actually tested different components of what makes up STK booster. For instance, STK booster is made up of beta glucans, 1,6 beta glucans. It's made up of epigalactins, which is from Camellia sinensis, that's from green tea. Now the beta glucans are actually extracts from mushrooms, and these compounds, they stimulate the immune system. They're able to provoke the production of interleukins and cytokines and all that. So it does that. So the mechanisms are all clearly defined from all these, from the substances that are actually making up STK boosters. Now, apparently, and this is very interesting, the STK booster is made up of 60% of these compounds. That is 1,6 beta glucans, epigalactins, and macantins. Macantins are also immune stimulants. So 60% of that composition is made up of this. Now 20% of it is made up of antiviral compounds. So it actually has antiviral compounds as well. And then 20% of it is made up of what? Antioxidants. So this is what make up this. So it's a very interesting and very powerful cocktail. So it boosts the immune system. Now, when it boosts the immune system, simply means that, of course, the immune system is able to fight and distinguish what is self from non-self, which my colleague would be able to understand that very well and explain that. So it has that prophylactic activity as well. But then it also has the antiviral activity, because it also prevents, because one of the things that it does, which we found out from one of the compounds, that it actually makes the cell to produce enough zinc. So it produces enough zinc, and zinc prohibits what? Viral replication and multiplication, which is very, very interesting. So those are some of the mechanisms that is there. So That aspect, it actually has some antiviral activity. It has the preventive activity all in one, and of course the antioxidant activity. So in a nutshell, that's what we've been able to come up with there. And right now we have a repurposing mechanism which we've been able to derive against COVID-19. And as I speak, it's been recommended for clinical trials across the entire continent by the African Union Scientific Research and Innovation Council, and also the Advisory Board for African Union. So we just finished writing the clinical trial package, which is about 180 pages, to run that clinical trial across the continent in collaboration with African CDC as well.
So, Dr. Minang, I mean, what's your understanding of, I mean, you can, as an outsider, listening to a colleague, what's your take on that?
Okay, so I'm back on now. It turns out my kids were using the internet downstairs. So somebody even told me I'm very clear now. Exciting work that Dr. Kenneth is doing, fantastic work. But this is the slight difference between what he's doing and vaccine research, and also where they overlap. Okay, so for vaccine research, the key word is specificity. You're trying to generate a response that's specific to a particular microbe. Okay, what he is doing, what their work is, it can be considered more of like getting a drug. When you already know that you have an infection, then doing a general boost of the immune response, you can clear whatever microbe that person has at that time. So you know somebody's already diagnosed with COVID. Then using a booster like that, because what that person needs is a good immune response, then you'll be able to knock out COVID. There are specific cytokines that are known to have antiviral properties, and there are some cytokines that are known to have antibacterial properties, and there are some that have antifungal properties. So, like in product work, knowing what agents that they have that tend to skew production of different types of cytokines, and knowing that what cytokines have antiviral properties, they can be able to tell you then that if this person is infected with COVID and we give them this compound, it will boost production of this type of cytokine that we know has antiviral properties. And so by default, it will likely knock out COVID. And then they can do studies now to show they have that effect. Way of last, they know very little. The little secrets of immunology is that they bring in a vaccine that has something that is specific to an organism, but they throw in something in there that's literally dirt, but that helps enhance the immune response. So you can imagine if we get a vaccine down the road that is really specific, all vaccinologists are very excited about the work that Prof is doing, because what they want to do is to combine those two. Because sometimes the vaccine will be even more potent in an inflammatory environment, in a cytokine environment, that because some of those cytokines, apart from being antiviral, are also significant immune boosters. Where if you already have a specific T cell or a B cell, that cytokine helps to enhance the expansion, the division, the reproduction, the multiplication of that T or B cell in the context of a specific pathogen that's already specific to. So there's no specificity there, but there's indirect specificity because you know that there's some cytokines that tend to have antiviral or antifungal or immune boosting effects.
So
sorry.
Bro, before you, I want to follow up on something, because again, I want to tie this to something that you had announced here before. There's, within the context of HIV, you have been, at least I was told, I don't know whether there's any data, clinical data, you have been able to produce some medication or something that is helping HIV patients in Cameroon. For everybody, they will think that they will say it's a cure. You have been able to come up with a cure. Now, this is, again, I'm trying to follow up with this the booster that you are producing. Is this something that falls along the lines of getting, call it a vaccine or a cure or both? Because from what Dr. Minang is explaining, vaccines have to deal with some specificity, and the booster that you have brought seems to have a little bit of it and a little bit of the general. Did I get that right? Or?
Absolutely correct. When you say a booster, vaccines are very specific because you have a target, you have a platform for delivery, right? So those things you cannot miss them. That's what you have vaccines. But then, of course, you remember I also mentioned about therapeutic vaccines, which is slightly against the concept as well, which Professor Victor Anomango of memory was. That's actually what his vaccine was built on, that premise that is actually treating. So therapeutic vaccines are also slightly against the context, slightly against the conceptual framework of vaccines. Well, like from what Dr. Minang has actually explained, again, the immunology is very clear. Provoking production of cytokines, which are antiviral, which are also antibacterial specifically. Yeah, that is clear. Now, what we are producing basically is not a vaccine. Yeah, it may have certain overlapping mechanisms of the way vaccines will work. That's for sure. But then, in its entirety, it's not a vaccine. All right. Vaccines are what you provide the attenuated form of the organism into the system of the person. That's it. And then what does that? What does it do? So that's like an antigen. So it provokes what? Antibodies now are being provoked. They produce the antibodies that are specific. I mean, if you look at the lock and key hypothesis. So they come in now and they make the system to be prepared and keep a memory in a lay way, in a lay term. So much so that when the person is challenged with that particular COVID-19 or whatever specific virus in question or specific organism in question, now, what happened now? That memory comes to play, and then the immune system sends signals for production of enough antibodies now. They come in and then they grab the stranger and they neutralize it. That's the whole, I mean, in a nutshell, explanation of what vaccines are doing all about. I think Dr. Mina would agree. But what we are giving here is not following that concept. It's not following that concept. Yes, it has some little attributes, but it doesn't follow that concept specifically. So, because we're giving a substance here which would provoke the production of cytokines, interleukin B, alpha, beta, gamma, interleukin. So, which are all protecting the immune system. But then it has antiviral properties. And then it also has substances that provoke enough zinc to be produced. And zinc actually inhibits the viral replication. And if you look at, that was the premise in which they were trying to explain the mechanism of hydroxychloroquine. That it goes in and then it provokes enough zinc to be produced, and zinc actually inhibits I think protease, or inhibits transcriptase enzyme production, and on account of that, frustrates the replication of the virus. So yeah, we have a conglomerate of mechanisms in this STK booster, if you will put it that way, and I'm sure Doc is going to agree with that, but not entirely that it's a vaccine. Now, on this, I wouldn't say more details about this, but I can share this. I don't want to put the cart before the horse, because there are some very intrinsic details that we don't want to put them. They are quite proprietary. Yeah. But what I want to say here is that probably, I'm sure AU would come up very soon with, because they've already, this was agreed by the advisory board of AU. It was agreed by the scientific council. It was agreed by the technical commission. It was endorsed by scientists across the continent of Africa. They have to endorse that. So you can imagine all the stages it has actually gone through to scrutinize this and get signatures that represent South, East, North, West, Central. It wasn't an easy process, but we all went through that. Now, but in the proposal for the clinical trial, the longer term is that we have proposed what they call a phytovaccine, which is a new concept. Okay, so we have substances that will provoke antibodies production, and then we shall harvest the antibody production, which are specific for the target organism, and then we shall use that to produce vaccines. Dr. Jacob, do you agree with that? Do you see some sense?
So move from something to
At that stage, we are trying to bring in more immunologists. This is what we have it as one of the strategies on our proposal, which AU. So we're going to be working on that as well. But then we have already identified the part in which we are going to. So we know what we're talking about. We know this. So it's not something you just jump into. We know what we're talking about. So we're going to work on that. All right.
Prof, let me get some clarification, at least for the sake of the audience. So you have a regular vaccine, then you have a therapeutic vaccine, and today I'm hearing something about is it phyto vaccine? What is the difference?
Yeah. What is a vaccine? let's start from the conceptual framework. What's a vaccine? We've just explained here. We've talked all the jargons about vaccine. Okay. We want our audience to follow. I want to, because when I came in, I followed what Dr. Yongabi. He has explained immunology here very, very well. This is clear. There's no doubt about that. But I want our audience, especially those that are non-medical, to be able to understand and be on the framework what we're talking about. A vaccine is what? A substance that we give to you to make you prepare for war. So eventually when the war is coming, we have an inbuilt preparatory mechanism. Then we can now unleash that inbuilt preparatory mechanism that we have prepared you and is lying docile in you, but will recognize you. And that recognition is specific. That means if we give you an inbuilt mechanism to prepare for an oncoming thief, it should only be for the thief. It's not going to be for any other soldier, whatever. It's only for its particular thief because it has the image of that thief in mind. All right? I'm trying to put it in that way. So now, when that thief comes, eventually, if another thief comes, forget about it. If somebody comes to provoke, which is not that particular thief that he has in mind, because he has the morphology, he has all the structures in mind, then it's not going to work. So that's why we talk about specificity, and that Dr. Jacob explained that concept of specificity. So it provokes immune response. The immune response with that particular target already produces what? The antibodies that has that structure in mind. And through the lock and key hypothesis, it goes in, clicks into it, and neutralizes it now. That's the way it is. So by going on this that we're talking about, no, we don't have a vaccine, so to speak. What we're having doesn't have a vaccine. It's not a vaccine, so to speak. But it has some tendencies, some characteristics of mechanistic functions that overlap with the way vaccines also function. That is number one. Now, he talked about a drug. A drug is what? It's entirely for treatment. You don't take antibiotics to prevent any disease at all. You take antibiotics to treat, and it has the mechanism. There are different mechanisms, maybe cell wall, inhibiting cell wall synthesis, inhibiting RNA synthesis, inhibiting DNA synthesis, nucleic acid, whatever. There are different mechanisms in which antibiotics they function. So it's going to treat. So this is not, antibiotics are not vaccines. Now, when we talk about a phytovaccine, what we're talking about here, and it's a new concept that we are working on, and we have actually identified what we mean by that phytovaccine. So we give you a substance. Now, that particular substance is meant to be able to provoke, of course, a booster. It boosts your immune system. You know, you get the point? Now, but then along the line, as it's boosting your immune system, because you can have a non-specific boosting of immune system. Dr. Jacob will explain this. Non-specific boosting. That, you know, that means it boosts your immune system, increasing the production of cytokines, which have antimicrobial activity. You get the point. Antiviral, antifungal, antibacterial. So that's quite generalized. Now, what we want to be able to explore is that when we give you that, it produces all these immune boosting cells in your system, but then we're going to harvest it from your system and then begin to see whether we can now have a specific target from that. It's a wild harvesting, and then we start seeing whether we have a specific target for specific diseases, coronavirus, whatever, other viruses, other viral infection. So we're terming that generally as phytovaccine. So it's still a work in process, and there are some intrinsic details on that. It's something we have also we already put on the proposal, and I'm sure that we're going to be bringing in a lot of immunologists on that. But that could be a different discussion for another day.
Thank you very much. What's your reaction to that, Dr. Minang?
Okay, so there's a lot of immunological information that is packed into what they are trying to do, and
exactly,
it's a new concept, and as you can imagine in science, I can already tell Prof that there's going to be a lot of resistance because what he describes doesn't answer the fundamental question in classical immunology, which is about specificity. It doesn't mean that you're not going to end up with protection. Saying that doesn't have specificity does not mean that what you're doing is not going to be helpful. But I see where there's going to be a lot of excitement in the immunological field among immunologists, which is this what I always call, well, immunologists call it the dirty little secret, again, as I keep on repeating. The fact that what you're doing could be used as an adjuvant. something that will boost the immune response in the presence of that vaccine that actually provides that singular thing that's needed, which is the specificity. And the specificity doesn't have to be the biggest thing in that context. So your Pfizer vaccine might actually be the biggest thing in that context, but just be able to combine it with something that provides the specificity in the response to make sure that your target is not wild and broad acting, but it's more specific and narrow. Because immunologists are more about specificity, and that's why a lot of times there's controversy about new concepts when you cannot really talk about specificity. That was the issue too, Professor Nomango's vaccine concept, the therapeutic one, because they don't imagine a vaccine being therapeutic, treating. They imagine a vaccine preventing an infection. And so his vaccine concept was based on people that are already sick, you getting the virus strain out of them, and then inactivating it in vitro in a test tube and giving it back to them. And you could see an effect. And the argument is not about whether you see an effect or not, which though that was the major distraction and it didn't go any further. The argument was that that's not your classical vaccine.
Yeah, that's quite good. You see, in science, you must it's all about fighting resistance when you have a concept. That is very clear in science.
Yeah.
And that is why those that were aiding Professor Anomangu did not help him either, because they could not wage the scientific fight and the scientific logic. And if you notice again the concept of specificity, you realize that what he was doing was that it was more specific. That means for 10 patients, you have to collect their samples, do the preparation for each individual. Exactly. So there was no one size fit all. Again, that's right. So it wasn't a one size fit all. So again, that's still attuning to the concept of specificity in immunology. Okay. Right. Now, on this very concept here, if you look at Africa, my brothers, if you just look at Africa and our vaccine, what is the innovative science that we want to be able to come? We want to be able to come up with an Afrocentric vaccine, isn't it? So, and we will push it through, because we're going to bring in all the immunologists and all the brains to sit down and re-examine exactly what is the premise of this, what is the science behind it, and so on and so forth, and then we move it forward. I'm glad for one thing you've said. You've looked at it and said, yeah, it's going to give protection. You've seen it with the lens of an immunologist, which is good, and I'm happy about that. But then you said, we're going to use adjuvants that are specific. That is good. What is the ultimate goal? The ultimate goal is that, is it going to work? Is it going to help our patients? Yeah. Beautiful. That is it. And at the end of the day, the end justifies the means. We'll say, yes, but that's the science. So that is it. And for innovation, we must make this breakthrough. That is the only lens I see that we're going to make medical science a little bit different from Africa. When we come out we speak what the whole world will say, no. And then, but when they come to examine it, they say, yes, it works. That's it. So, just supporting your thoughts anyway.
So, I know we have
and you are welcome. And you are welcome to join that effort eventually when it's getting ripe. Dr. Jacob, and of course Lamed is already there, because I think he's already being the one propagating and putting this on the media, which is okay. So you're welcome.
So, Doc, again, Prof, I wanted to say, we are talking about Afrocentric solution to COVID-19, and you just mentioned this. I'm wondering where are we with the remedies that came in from Douala, with the Archbishop of Douala, Bishop Cleda, and the one from
Madagascar.
And where do you situate these initiatives with what you are coming up with?
Yes, actually, our work cuts across the 47 member states of Africa. AU will recognize 47 member states. So we have the letters, we have the mandate to do that for the 47 member states of Africa right now. So what we are also doing is to examine, we have the terms of reference to examine all the traditional medicine claims that are going on across the continent. All medical claims, because there are also medical claims going on. What I mean by medical claims, like, for example, if dexamethasone, for instance, instance, has been claiming in Africa that it treats COVID, which I doubt all the same, then we have to also examine that claim. All right. So we are examining all that. Right now, we would have a center. Cameroon represents the center for Central Africa. So on our team, Cameroon represents Central Africa. Nigeria and Ghana represent Western Africa. Namibia represents South Africa. Egypt represents North Africa, and Kenya, Tanzania represents East Africa on this committee. Now, with what Bishop Cleda is doing, these are anecdotal claims whereby the science is not there yet. Well, the science may be latent, but for now, we don't do science and clinical science that way. That's not the way to do clinical science. Now, he has used his position as an archbishop to do that. If a scientist had done that, he would have been in jail by now. So he has used his position as an archbishop to make that claim. And the case of Madagascar, which also came up, is something we're also putting up in our committee to also examine. He used his powers as the head of state, he's not a scientist, to make that proclamation. If a scientist had done that in Madagascar, the scientist would have been on total global condemnation at the moment today. That is very, very clear. Just imagine Dr. Minang coming up, in the global world to say he has tested this. They will ask you, what is the scientific protocol? What is the clinical trial that you did? What are the ethical approval that you went through? Who examined all this? So all those things will push you into jail. So these are all anecdotal claims. And what we are trying to do here is to be able to, advise that the process, the due scientific process should be followed. Yes, people are going there and taking their treatment, whatever treatment it is. But what are the clear diagnoses? Even now, there's a testing controversy on COVID-19 across the continent right now with the testing protocol that are being used. So you may have a lot of false positive cases and false negative results for COVID-19. You cannot use that to be able to adjudge herbal concoctions like that. So in a nutshell, what they are doing is very unscientific. We are happy that they are trying to look for indigenous solutions to problems. And if you look at these people who are doing that, well, he is also a traditional traditional doctor, all through his life for quite a long time. But there are many traditional doctors too in the continent who will claim that they can do that, but they don't have the audacity and the position like they have to be able to make such claims to as well. So we need scientists on the continent to valorize these claims. What I have now, it has taken 17 unbroken years to develop STK booster and STK phytoimmune that is going now on a clinical trial. We did toxicological analysis, huge ones, a lot between in vitro, different in vitro toxicological analysis. We looked at the effect on liver, the effect on kidney, looking at all the liver enzyme profiles, creatinine, all these profiles were done. We got results. Then we looked at efficacy, antiviral activity using, different cell lines. We've done that. We have some results. Then, of course, we have the results from the anecdotal claim, which we call the exit poll clinical analysis. Which is the exit poll clinical analysis is the Afrocentric. Afrocentric in the sense that people will come to you. They don't know whether you have done clinical trial and whatever you have. They just know that from ancestral knowledge, this person is good at this. So we call that an exit poll clinical kind of trial. We got that result. Then we file in to Food and Drug now to examine our claims. The Food and Drug in Nigeria, that's NAFDAC, went through for a period of 1 year and then had to ask for all the samples of our claim. We provided them the samples. They did their own independent study and came up with a 250 pages volume of all what we had done as claims. And then they had to now compare with what we have done and what they have done. And at the end of the day, they gave us a license, what they call Food and Drug approval. That's a NAFDAC approval. They gave that approval. Now, the National Institute for Pharmaceutical Research, which is also a regulatory body in Nigeria, had to come and visit the production protocol to see where the production is. Does it follow the international standards? Does it follow GM, good manufacturing practices? So they have to be able to ascertain all that. and they look at that. So that is on account of that we compile all this. We have the evidence of this, in order now to file in for what they call a clinical trial, because we've done enough of preclinicals, and we have all this. We've got a license number, we've got approvals from all the licensing body, and then now they have to set up different scientific committees to examine all this, despite the fact that we've got license. The scientific committees across the continent, so they have endorsed through a letter to endorse all the scientists representing different different regions of the continent. They've endorsed that, and then they move now to the scientific council. The scientific council had to look at that again and the endorsement. Then I had to do a 2-hour presentation to the scientific council again on that. After looking at all the documents, now they endorse that. Now, when they endorse that, they send it to the Scientific Advisory Board for African Union, which is the topmost. And I must be happy because on the Scientific Advisory Board, we have 3 Cameroonians who were also there. Professor Beban Samin Chumbom, who is the president of Cameroon Academy of Science. All the presidents of Academy of Science of Africa were present. We have Professor Jean-Claude Mbanya from Yaoundé University Medical School, who was also a member of that board as well. And Professor Charles Wisingong Shea from South Africa was also partly a member of that advisory board. The advisory board looked at it. We had a 3-hour defense, and to examine all the reports of all the different councils that have met on it. And then at the end of the day, they unanimously now approved for an AU continental clinical trials now on the product. So that is the way to go. So if they have these claims, let them go. They have to go through all these processes as well. Okay. So
yeah, but my problem with that is if they have to wait and go through all that, many people would have died before they ever go through all that. And I think that's the claim for many people. What works, if it works, it ships, isn't it? To paraphrase.
Yeah,
I understand that the science is very important. And yes, Dr. Minang, you wanted to say something.
Yes, so I want to particularly thank Prof for that comprehensive summary, especially about your unique position and the organization that you had in Africa, and the comment that Lambert just dropped there goes to the heart of the debate that we have with our fellow scientists in Africa, or just the average African layperson that thinks that we African scientists, we are usually the first to stand in the way of the potential for an African to make progress, and you find it very hard getting through to them. So I'm really excited that you're able to summarize it like that. We know that we have our African traditional treatments that we've had with us for millennia, but trust me, it's not only Africa that has traditional treatments that have had for millennia. Every group of people have had traditional treatments for millennia. The Indians do, the Russians, the Coptics, the Europeans. But that's not science. Science is removed from that stuff. Science has standards, has protocols. Okay? We know that when you're a dying man, you can grab at a straw. That is true. Even the United States is going through that. When 2,000, 3,000, 4,000 people were dying a day in New York, every physician in the hospital was willing to try anything that somebody mentioned that it could help. You're just trying to save someone who is dying. But that's very different from the science of proving that something works universally, that you can start giving out there to other people that are not on their dying bed yet. They just happen to be infected. And that's where science comes in. So when you go out there and you're claiming that something can treat, and you're putting it out there for everybody to know, and that they are welcome to come and get a treatment when they are not even in their dying bed, the risk is that might be poison even. Some people that would otherwise have been fine, even though they had the infection, because not everybody that's infected with HIV that will end up dying. There are a few people in the world that actually control the infection. So I'm not about to take poison if my body could control it. But I'm willing to take poison if I'm in my dying bed because I don't know what is going to help me. We have to separate those two out. So the emotions a lot of time is usually when people just say, oh, but these people are dying. Why are we waiting for all the science to come in? Fine. A physician, just like that traditional doctor, actually is permitted. In the US, that's what they call off labels, the FDA. A physician is allowed by their license to make that snap at that moment to say somebody that will die otherwise. But when you're trying to come up with something that you can use as a preventive for the general population that may not die, but they may end up with debilitating effect, in their life, that might affect the quality of their lifestyle down the road if you do not treat early. We want something that has gone through a rigorous testing process, that is open source. Even though we talk about proprietary, the pharmaceutical companies that hide information about how they come up with their treatment, they don't hide information about how they test it. It's always open source, so everybody can know that this is the number of people that we test in this number of continents. This is how sick they were. This is how we categorize them. This is how we did a randomized, double-blind, placebo study. So just come up and say, oh, and so what we fear is, like Prof mentioned, because somebody is an archbishop, which means they have a very loud microphone, they tend to sell us snake oil, when maybe a herbalist in Bambili, my small village, may even have something better, but they don't have that loud microphone, or could be selling the same poison because they don't have a loud microphone. So we have to be careful about people that have loud microphones and are selling us stuff for general use.
Good. So again, this is a good way to get into the next thing that I talked about in my intro, which is about controversies. Okay, because we all know about the anti-vax movement. There has been a systematic attempt to say that vaccines are dangerous. For example, I know one that is very prevalent is about the depopulation myth. So vaccines are created in Europe in order to depopulate Africa. And so we should avoid that. And I'm sure you all listen. I don't know whether you followed up. There was an audio message that was circulated this week claiming that is the voice of the president of Ghana reading out some message from Rockefeller Foundation, giving a roadmap of how they have planned this thing long ago, meant to, just scare everybody. And it's quite interesting. So I want you to take your reaction to this. Are vaccines that safe? Can we really trust that these vaccines, this mad search, this mad rush for vaccines, is that something we really have to pay attention to?
Prof, should I take that?
Yes, please go ahead.
Okay. So are vaccines safe? Yes, if you are referring to a vaccine as a final product, okay, that has gone through all the hoops that Prof was at pains explaining. That's why we have to be careful when people that have loud microphones or a loud platform are pushing something. Okay, it might not be a vaccine, it might be a claim for a treatment or a drug or whatever. When you're pushing things that have not gone through that rigorous testing process, then it's very unsafe, it's very dangerous, okay, and it's irresponsible. But vaccines, as the final product, are very safe, because for them to be a vaccine, and that's why we're giving this timeline of 2 years or 5 years or 10 years, the bulk of what goes into vaccine research is not finding out if it's immunogenic in the sense that it can elicit an immune response. No, it's actually finding out that it is safe. Safety studies, there are four levels of clinical trial, step 1, 2, 3, and 4. Is the last step that's finding out if a vaccine can do what we intend for it to do, which is protect against an infection. The first two and part of the third step is finding out that it is safe. These are safety and immunogenicity studies, and those studies start with single cells in the laboratory, then move on to small mammals, then large mammals, and then before the human population, and then make sure you classify them correctly. And then, but we still know that at the end of the day, you don't get 100% safety, but you have taken, but only ones that have a significant degree of safety end up crossing the mark. It's the same thing with the drugs, like chloroquine. They keep telling you that it gives people that they give heart arrhythmia, all of that. But that's only how safe it could be. But before that drug came into the market, the biggest focus was the biggest focus to find out if it's not toxic in the first place. That's the biggest part in any drug and vaccine research, as Prof will confirm to you. It's about toxicity. That's why immunologists or people that work on drug research, they recall and they are lost for words when people are trying to push a certain compound or substance to mass use when there have been no safety studies or toxicity studies, and they're just saying, oh, it works. And then when people try to push back and say, oh, you people, you're not supporting your African brother, or you don't like things that are Afrocentric, you think only the white man is better and all of that. No, people's concern is about toxicity. It's not about if the thing actually does what we're aiming for it to do, because that is secondary. If you drink bleach, It will kill every organism that infects you, right? But it's going to kill you as well. That's why the whole country was up in arms when Donald Trump said that we could use Lysol or UV rays against the virus. Of course, it's going to kill the virus, but guess what? It kills every living organism.
Yeah, I know.
Okay.
You have 1 more minute, Dr. Minang, in order to go back. Prof, can you respond to that?
Yeah, I think he's addressed the issues. For any search for a vaccine or a drug, what is central, like he has said, is looking at the toxicology. That is the first 2 stages, is to be able to ascertain the toxicology. In fact, with STK boosters and STK phytoimmune, we spent the large part of the research was looking at toxicology, subchronic toxicology, looking at all the organs. How does it affect the heart? How does it affect the kidney, the liver? So we looked at all that. So with vaccines, it's the same procedure. The key thing for any of such to be licensed and in the process is to look at the safety. Now, once the safety comes first before efficacy, because normally they would anticipate, like he has said, is that if you drink bleach, it's going to kill. Bleach is effective in killing corona, but it's going to kill you. Yes, okay. So it's effective in killing the coronavirus, but it's also going to kill at the end of the day. So there's this concept of selective toxicity. It's very critical. So that is that. So now on the issue of all the conspiracies and what is moving on, really I find myself sort of really lost. What I do in such circumstances is to be able to filter the science that is there. I don't want to be caught up in the web of politicians and older, social advocates left and right, because I don't know which agenda they are pursuing. There's a lot of work for scientists to be doing right now at the moment than caught up in that web. I know that it's that kind of confusion and conspiracy. It's all about money and funding. It's all about fame. So for us at STK, with what we've been able to develop, we stop focus to what we are looking at. The scientific procedure for discovery is very clear. It's like a constitution of a nation. It's like a statutes of an organization. You just have to follow it. So I would say that let's not be distracted with all these issues, because I don't know what agenda they are trying to follow up. And for me, I'm not sure I have enough evidence to do all that investigation, whether there's an element of truth in that. But I think it's a distraction. All we now have is that corona cases in Africa, they are spiking all across, which incidentally and consequentially, what is that telling us? That the mechanisms that we have in place are not working. They are not effectively working. Either the testing mechanisms that we have in place, or we are not testing enough people, or that the measures that we have, they are not really working, because as we speak, the values are increasing in Nigeria, they're increasing in South Africa, they're increasing in Cameroon. And of course, we have a resurgence again in China. Look at what is the catastrophe in Brazil. Yes, it's quelling down in the US, but the colossal number again in US is still worrisome. So that's for me where the focus right now is. The conspiracy issues are all about fame and who is in leadership, who is in control, and who is in control of money. So we leave that to those people who want to spend their energy on that. Let's focus on how we can be able to develop this. Now, on a very final note for me, I would say vaccines, yes, across Africa. How many of African scientists are working on vaccines right now? Just today, a Nigerian Dr. Olowale claims he got a vaccine for corona from the University Adeleke University in Nigeria. Just forgot the news today. So again, these are claims. There are so many very cheap claims. But then if you want to look at the science, my brother, the science takes the time. If you want to go through all the different steps, it may be so frustrating that it will be easy for you to make a cheap claim. I'm sure you know that, because you want to divulge the scientific procedure. So I think in Africa, I would propose that while working on vaccines, we should also work on so many therapeutic techniques, strategies that we can also put them down, pari passu, to be able to carry them along. Let's not just focus on one intervention, because vaccines are going to take a while. And even drugs, effective drugs, are also going to take a while. We are just lucky that STK, 2 patients from UK took it and they recovered from COVID, but that was an accident. And then that gives us an insight. And apparently these were whites who had they got this product from Douala, and while in UK, they caught COVID-19. And again, like what Dr. Jacob also said, in a quest for survival, they took it. It was not meant that they were going to take it, but in the quest for survival, they found out that the only option for them was, let us try what we got from Cameroon. When they tried it, they recovered. And now they are asking for 12 cartons of it in UK. They wrote us to ask, people are writing to ask for 12 cartons. And no one is asking us. You would expect someone from Britain to start asking you about safety and toxicity. But they have seen a case there, 2 of theirs that have taken this and recovered. So that gave us an insight to be able to repurpose, because initially it wasn't licensed for COVID. It was licensed for what? HIV. And it was licensed for herpes and hepatitis viruses. Okay, so we now have to look at the mechanism and try to see how we can repurpose it for corona COVID-19, and that is what we've actually been able to come up with. A systematic, analysis of it, and then of course that's what is being approved now for us to recommend it for a clinical trial now. So going forward, let's look for holistic scientific approaches. Vaccine being just one of such interventions, but there are other interventions that we should carry along as well. Thank you.
So thank you very much. But before you go, I'll come back for one last thing. But Dr. Minang, to you, your last word, and I would like to ask your last word with respect to, let's be a little practical. I know we've been talking about vaccines, discussing all this. You have relations in Bambili who are struggling with this. What advice are you giving to them? And what advice are you giving to young men who are in school or who are out of school struggling, and maybe they want to venture into this kind of things? What should they be doing in order to prepare for a career in immunology?
Okay, so 2 things. So first, on the COVID side, let's do what works. We know that social distancing works, personal hygiene, infection control, wash your hands frequently. Don't touch your face, your eyes, or your mouth with your hands if you have not washed it between interactions with people or surfaces. It's that simple. It's very easy. We touch our face a couple of 100 times an hour. So if you can reduce that, or make sure that between the times you're touching your face, you're washing your hands, you should be fine. That's it. About immunology, it's a very exciting field, and there's so many things you can do as an immunologist. Unfortunately, you don't start up studying immunology directly. You have to go to secondary school, high school, do the sciences. But even if you did the arts, that's not really an impediment. You can still take biology courses later on and take supplemental courses. But the key thing is you want to be very grounded in chemistry, biochemistry specifically, and biology. If you have very good grounding in those 3 areas, you should be good to go. There are other additional things that you can do as an immunologist. If you have an understanding of molecular biology, if you have a general understanding about different infections, different pathogens, the epidemiology of disease and pathology of disease, then you're good to go. But it's a very exciting field because you can get into vaccinology, you can get into biotechnology research, you can get into pharmaceutical work, and so many things. Even if you get into medicine as a medical doctor, if you have a background in immunology, it really shapes and informs your practice.
So thank you very much. Don't go away. Just one more. I know you are pressed for time, but Prof.
Yes.
The last time you were on this show, you gave us a little. I don't want to call it a proposal, because at the time people were taking the steaming and then taking garlic and all that. And you gave us a little thing there. But my question to you is, when should people stop taking ginger, garlic, and this mixture? Is this something that has to go on for eternity? What should people be doing now at a practical level to fight off, ward off, while the science is exploding?
Okay, just a minute, please. Let me just take a call. Yeah, hello, sir.
Yeah, Dr. Minang.
Yeah, I'm okay, sir. Yeah,
Let me
finish up a call.
Leave now. It looks like Prof is actually taking a call.
Yes, I just muted him because I was taking a call. But again,
so can I log in now and you can wrap up with Prof, or is Prof back?
Yes. So again, I just wanted to say thank you to Dr. Minang. Dr. Minang and I I was with his wife 2 weeks ago. We're here talking about education and COVID. Dr. Minang and I went to the same alma mater. We went to Bishop Logan College. We actually were schoolmates, and all three of us happened to have gone to Sacred Heart at different times. So it's a small world, and we keep getting together and getting better. So thank you very much for the time, and I'm sure we'll come back to this again when there's any news about the vaccines.
Can I come back? Can I come back quickly before he goes? Can I get to it before Dr. Jacob goes? Yeah.
Now go ahead, Prof.
Yeah. Thanks. Sorry for that interruption there. Dr. Jacob, for Lambert, it's a pleasure. I mean, when I got to Sacred Heart, I think, if I remember, just now looking back at history, I remember that Lambert was the one who showed me the athletics field because I was looking for the athletics field. So, I remember now. Then I said, well, let me bring it up to you so that he knows that he was one that showed me the athletics field in Sacred Heart College as well, and then even gave me a ball to play. So thank you about that. Now, and then now for the advice, it's not just taking ginger or whatever, because it's been it provides protection. You need, everything has a dosage. You don't just, if you overeat, it creates another problem as well. So I think people can now have a device and seek medical advice also. You take ginger twice a week, fine, because you are taking it, you just want to take it to boost your system, to be able to help you prevent. So it's not, you're not taking it, some of these herbals, we claim that they are safe, but, intrinsically, they are not safe as we think. To a certain level, they are not safe. People have been taking turmeric, but turmeric too is also dangerous. It has some side effects as well. So we need to be very careful. So I would advise people and say, have a schedule where you take these things very carefully, maybe twice or thrice a week. Take something like garlic, alum sativum. Alum sativum, when you take garlic all the time, you know what? It decreases your white blood cells. It actually causes hemolysis. It causes aplastic anemia because it lyses the red blood cells and causes aplastic anemia. But this is information that someone in their homes and on the street doesn't have this kind of information. So you need to be careful. So take it once or twice a week. Every week you have that regular schedule of taking it once or twice a week because you are just taking it as a prophylaxis. Yeah, that goes. So that's my advice. Not just taking them as if they are foods where you just take them all, in perpetuity. So you need to be very, very careful about that. I think on that note, that's what I would rather advise people. And some people even take it together with synthetic drugs. These things, there's a lot of incompatibility, even at the level of, even at systemic level. So people need to be very, very cautious. There is really the imperative to seek for medical information on these kinds of issues. Now, on issues of the advice Dr. Minang was giving to people, you see, we query social distancing. I would tell my people query. We came up. I consult for WHO and I query that. I query that a lot because when you are doing what they call risk communication, because that's what you're trying to do. You're trying to communicate risk to people and risk mitigation, and it's something we have also sent that to CDC on the kind of knowledge management strategy for COVID-19 in Africa should be rearranged. You say social distancing. When people live in their houses in Bambili, sometimes there are four people in a small kitchen, and they sleep in that kitchen, and then you are telling them social distances. How does that work? So we have to be more practical about that. Yes, tell them social distances. That's the message. But what does social distancing means to somebody in Cameroon, in Africa, when they living in conditions, the socio-cultural life comes to play? So we are working on practical modalities on how to practice social distancing in Africa within the cultural context. Again, that's the Afrocentric aspect of it. Now, washing hands, Dr. Jacob, what kind of water are they using to wash their hands? They just pick water around to wash their hands? Even the washing units that we have, they have water with high level of coliforms, water with contaminated water, and that is polluted. Then they wash their hands, and then just to be able to infect themselves with rotavirus or whatever, still, because rotavirus is resistant to chlorination in water. So we are also warning them and said, hey, you need to look at the portability of your water in which you are using to wash your hands as well. So we give that extra message to them. Again, now sanitizers. What kind of sanitizers are you using? What is the standard operating protocols for using standard sanitizers across Africa here? You tell them to go and use sanitizers. How many people I look at COVID to take quite a while for it to be off the table, global table. That means people will have to be buying sanitizer. And I'm sure if you want to talk about COVID compliance strategy, it's not going to work with so many people in Africa. They would get tired about using sanitizers because they cannot afford it all the time. Now, what are the protocols for using hand sanitizers? It's made of alcohol. You see terrible sanitizers these days having alcohol, and they inhale alcohol, which is carcinogenic, right? So again, we need to be able to tell people the right messages. The risk communication strategies must change. We must have what they call, strategic, fit for purpose risk communication strategies in Africa must be like that, because alcohol itself is also a carcinogen, and we need to be very careful about that. Water too, we must think about the kind of water they're using as well. So again, these are the things that we need to look at them, and that's what our working group, research group, is looking at all these strategies. Even the face mask, people are just using all kinds of cloth to sew face mask right now at the moment. What is the porosity of this face mask? Coronavirus is between 30 to 160 nanometer in size. So what is the pore size? What is the filter size of the face mask that we're using? So these are things that we need to be able to look into and know. Not just saying we are put on a face mask. Okay, the distance between one person and the other is 2 meters. Who says you cannot have an aerosol that can travel even 5 meters? So again, these are some intrinsic challenges. We developed these techniques caught up within the heat of an epidemic, of a pandemic. So there was no proper research specifically that looked at the significance of all the intervention strategies that we're talking about. So when we give them, we still give them with care. That yes, don't rely totally on this, but this is a remedial method that we're going to have now. Use that. But while we're working, on looking at that, who says 2 meters? Somebody woke up and said 2 meters. What is the scientific basis of 2 meters apart? So again, these are the challenges that we're talking about. So while we are taking that as remedial methods, we are still busy working. All right. Thank you very much. It's been a pleasure, Dr. Jacob. It's been a pleasure, Michelle Lambert. I really appreciate you, and I really appreciate the work that you're doing. And we'll keep in touch.
So thank you very much, Dr. Minang. Thank you very much, Professor Kenneth. I really want to say thank you for offering your time this Saturday. And I'm looking forward to more of these interesting conversations to help the community to improve our knowledge base. And I hope that, Dr. Minang, you're going to pair up with Dr. Yongabi so that we find out more about this phyto vaccine, which is what we came up with today. And for the sake of those still watching and listening, vaccines, my basic understanding today is a memory card. It's given so that memory, when the thing attacks you, then is able to fight that off. And while the search is going on, there are little things that each of us in our little communities should be doing. And let us always search, look out for knowledge. Let us not just use things because they are available and they might have worked for one person and they might not work for you. We continue to help each other. Thank you all and have a good day, the rest of your Saturday and a great weekend. Happy Father's Day and Juneteenth, and happy Refugees Day for our refugees across the world. Thank you all. Thank you, and see you next week.
Thanks.
Thank you very much. My pleasure.